Good morning, all, my dear students, colleagues, and other faculty and other aspirants of listening to this impact lecture titled as Artificial Intelligence Driven Advances in Metabolomics Unlocking the Secrets of Disease Diagnosis. So, I introduce myself as Dr. Kiranmayi Mandava, Principal and Professor, St. Paul's College of Pharmacy Autonomous, and this institute is located in Hyderabad, Telangana. So this impact lecture is under the flagship of Institutions Innovation Council, St. Paul's College of Pharmacy. So I'm thankful to IIC, Ministry of Health and Ministry of Education for taking this great initiative from half the decade time to initiate all these research and innovation oriented programs to inculcate research innovation ecosystem in every institution. So with this brief introduction, I would like to share my slides. So today I'm going to focus on the main keyword metabolomics. So for many of the academic researchers, metabolomics is not the familiar word actually, and it needs extensive research. So for that reason only I want to introduce this word and also what and all happening in the world in and around the academic institutions or premier research institutions in the world, what kind of research is happening in metabolomics and what is the present era of metabolomics, need of this study, and what are the key components of metabolomics I would like to discuss today. Coming to basic definition, actually metabolomics is the youngest of omics triology, and it is defined as both qualitative and quantitative investigation of the entire metabolome of a biological system, has positioned itself as an indispensable methodology to investigate global biochemistry phenomena at a cellular level. So to be elaborate, omics triology is nothing but, omics is nothing but it's suffix of genomics, proteomics and metabolomics. All three words ends with this suffix omics. All three are involved in this in the present era of research, so we are using the word triology. So metabolomics involves both qualitative and quantitative investigation of the entire metabolome. What is meant by metabolome? Metabolome is nothing but a collection of all the small molecules in the cells produced by the cells of a biological system, has positioned itself as an indispensable methodology. It's a very important word to investigate global biochemistry phenomena at cellular level. So biochemistry we used to study at our graduation, post graduation level, our research point of view also we know biochemical pathways and all, but biochemistry at cellular level in the research, so establishing the biochemistry at cellular level is nothing but metabolomics. So in other words, metabolomics is a multidisciplinary research because the present Indian central government also they call for multidisciplinary research as per the national education policy. So I'm feeling proud to announce this particular word because every discipline of life sciences now need to orient towards multidisciplinary research. So this metabolomics is one of such multidisciplinary research field involving convergence of biology, chemistry, chemometrics, statistics and computer science. It itself shows that it is multidisciplinary. We need a team of people with expertise at both life sciences and also statistics and also computer science. Okay. And to move forward, it can provide unprecedented in-depth explanation and insights of the mechanism responsible for various physiological conditions given the innovative developments in analytical technologies, advancements in chemometrics, statistical methods like big data management, big data analysis, and integration of orthogonal biological approaches, etc. So this is about metabolomics, definition of metabolomics. And coming to era of metabolomics, the metabolomics analysis originally emerged in the year 1999, proposed by Jeremy Nicholson. He is the one who initiated this analysis. This tool was initially described as a comprehensive analysis. Try to understand what is comprehensive analysis, comprising the identification and quantification of as many metabolites as possible in a biological system. So it's a very complex, biological system itself is a very complex one. See, how many, suppose if xenobiotic is introduced into biological system, so it with the effect of the organs, especially liver, it's an enzyme machinery. The liver produces a fleet of enzymes which helpful in metabolism, biotransformation, detoxification, and all. We all know about all this terminology. So after this effect, how many metabolites are forming at cellular level? This xenobiotic or food, whatever foreign substance which is ingested by the human being. So how many metabolites are producing? What about their concentration at cellular level? Of course, it's a very complex phenomena, right? So that's why metabolomics is the need of our awareness. Like we need to know, at least if we don't have facility to do all this research, one should know about what is happening in the world and what is happening in the premier institutions or the in other abroad universities or the research institutions. What is the status of metabolomics, metabolism, like metabolomics research, and what is the application of it, and how to procure the research facility to perform these metabolomics so that we can avoid a lot of adverse drug reactions. So I'll be covering all these points in the future slides. So coming to the comprehensive analysis, so you need to understand two words like metabolomics covers both identification, means qualitative, and also quantification of as many metabolites as possible. I'm not here in a position to mention these many metabolites because in biological system we may not mention it as possible in a biological system. It is a qualitative and quantitative and dynamic study of low molecular weight molecules. Typically, the limitation is less than 1500 Daltons present in organism, tissues, or cells at a specific moment and under a particular environmental condition. And what is the scale? What is the scale of metabolomics? So greater than 40,000 annotated human metabolites and an estimates are possible, like estimations are possible, and also highly diverse structural formulas and high number of isomers are also analyzed, and highly diverse physicochemical properties can be uh, studied and elaborated by using this metabolomics. This is the scale of metabolomics. And coming to health and disease, what is the correlation between metabolomics and health and research? Because I am pharmacist, so I am academic researcher. So as an academic researcher in pharmacy field, I want to correlate metabolomics to health and disease. Though it is multidisciplinary aspect, so every discipline people, they want to know to what possible extent we can do metabolomics in our sciences, like in our pharmaceutical sciences. That's why metabolomics in health and disease. So it has several applications in health and disease, including, especially I have highlighted the keywords precision, personalized medicine, single cell, epidemiological population studies, metabolic phenotyping, metabolome wide association studies, precision metabolomics. This is very important, and in combination with other integrative omics. I already mentioned what is omics. It is the suffix of triology like genomics, proteomics, and metabolomics. Okay. And coming to single cell metabolomics and single cell lipidomics, technologies allow high dimensional characterization of individual cells, disease heterogeneity. Heterogeneity means we know it's a complexity. It is not homogenic like similarity. So a lot of mixture of conditions will be there. We need to align with dots, all these things, in order to understand what is what kind of disease it is and what kind of biomarker we can use to identify or diagnose this disease. Complexity, identification, expression, abundance of disease associated small molecules, metabolites, and by LCMS, MS, GCMS, MS, live single cell mass spectroscopy, we all know that it is LSCMS, imaging mass analysis of various cancer samples at the single cell level reveal cell-cell interactions, especially tumor is concerned. it itself is a heterogenic. Lot of reasons are there for tumor formation, and this complexity can be understood by using metabolomics and by using advanced hyphenated analytical tools like this interface technologies, LCMS-MS, GCMS-MS, and LCCMS, etc. And moving to its what kind of strategies are there? Metabolomics is an integrated word. So we want to divide it into different aspects for easy understanding. So the main approaches of metabolomics can be classified into two: targeted strategies and untargeted strategy. Coming to targeted strategy, it's absolute. Hypothesis based it is, and quantification is absolute quantification, and small number of metabolites only we can analyze by using this strategy. And MSMS correlated to reference standards, it is possible, and it has limited applications. Whereas coming to untargeted strategy, the main two keywords of untargeted strategy is it will go with fingerprinting and also profiling. Okay, exploratory and generates a hypothesis. This is very important. Exploratory and strategy, untargeted strategy of metabolomics is exploratory and generates a hypothesis, and it involves with or without identification and quantification. So we can call it as a relative phenomena. Large number of data sets are possible, and MSMS correlated to databases and libraries. We know combinatorial sciences, combinatorial library and all, already database is available. So we can correlate the result obtained in the analysis with the databases, and wide applications are there for untargeted strategy. And I want to focus on untargeted metabolomics because they are relative, they are hypothesis generated, and it has wide applications. So this is the flow of untargeted metabolomics right from collection of biological samples from the human body, and data acquisition, it is very important. So what kind of data, like analytical tools we can use for data acquisition? For example, NMR, nuclear magnetic resonance spectroscopy, LCMS, GCMS, and we can generate the spectral data. And of that, spectral processing. This is very important. So anyone can run the data, even technical person, those who don't have subject knowledge, only basic technical skill he has, uh, he can run the spectra and he can generate the spectra. But interpretation is very important, and correlation, analysis of the data is very important. So spectral processing, like baseline corrections, noise filtering, peak detections, peak alignments, normalization, and deconvolution, these are very important. So feature matrix of the samples can be generated from here. And then coming to data analysis, as I mentioned just now, univariate analysis, whether we are going for univariate analysis or multivariate analysis, and supervised. Here also unsupervised and supervised multivariate analysis are there, two types, and multivariate method, and candidates biomarkers classification models. Here we need to adopt this. So biomarkers can be identified. You can see some like dots here, the identification picture will come like this, and metabolite parallelly simultaneously metabolite identification. So from the spectral database, dataset features and identified metabolite structures can be generated from this. And finally, altogether, in combination with these two, data analysis and metabolite identification. In data analysis, biomarkers can be identified. In metabolite identifications, structural illustrations can be possible. Overall, biological interpretation, nothing but pathway analysis. Okay, over representation analysis, quantitative enrichment analysis, single sample profiling, correlation based network analysis, Gaussian graphical modeling, network topology measures, etc. So with the help of all these tools, pathway analytical tools, we can interpret the biology at the end. So what? So in correlation with these metabolites, like suppose metabolite has been identified. So then that interpretation has been done. Then only we can come to one conclusion. So this is the analysis workflow in untargeted metabolomic study. Moving on to next slide. So I mentioned in the title AI driven. So what is the role of software here? Artificial intelligence here. So AI made these metabolomics very easy. Like we can run the show in such a manner, in a smart way to generate or to interpret or to come to conclusion about the biological metabolites and their interaction, concentrations at cell level, including structures. So here I have just listed out few softwares, tools used, and what is the type of the tool, and what is the target. And I'll mention at the footnote what is 127, 1225 and all, and what website you can refer for these tools, and what is the reference like author said, and which year they have published this data, etc. So for example, Metabo Analyst 2, type is web, target is MS and NMR, and features 1 to 7. Website I have mentioned here, Zia et al. 2012. Okay, and come, I'll just show the footnote also. So this is the continuation of table continuation. Batman is another tool. R is the type, NMR target. They need to perform NMR 3 to 4. Website Hardal 2012. So if you see, this table provides a complete and updated list of open source software. These are all open source softwares that is commonly used in the untargeted analysis of metabolomics. This column refers to the feature included in the tool spectral preprocessing. What is one? Spectral peak alignment, peak detection, baseline corrections, pathway visualization, pathway analysis, 2D NMR analysis. Likewise, they have mentioned 8 features. So out of 8 features, how many number of features this software can fulfill? For example, Batman. So it is fulfilling 3 and 4. What are 3 and 4? You have to see here, the footnote. Okay. And for example, Met Explore, it's a web tool, 6 to 7. What is 6 here? Pathway analysis. What is 7 here? Pathway visualization. So what kind of features they are going to cover, I have mentioned here. And then now, untargeted metabolomics means fingerprinting, profiling. It's a relative, and lot of data sets will be can be generated, and wide applications are there, and various tools we have seen, and recent advances in targeted and untargeted metabolomics. Suppose if we want to generate interest to learn something, means we need to know about applications. Why should we learn? So because there are several advancements, there are several applications, and how it is useful will be helpful for us. That is also important. So by leveraging the advantages and disadvantages of metabolomics analysis, so one can guess here what is disadvantage means complexity and little bit cost involved in it. How even little bit intellectual skill set is involved in interpretation. So team of people, especially it is multidisciplinary analysis, team is required for that to come to an endpoint. So this is called disadvantage, we can call it as. But any researcher need to leverage the advantage and disadvantage. So researchers have begun to develop strategies and combine multiple analytical approaches to mitigate and pitfall of both the techniques, both targeted and untargeted. For example, one study examining the metabolome involved in the pathogenesis of hyperuricemia utilized untargeted metabolomics to screen novel candidate biomarkers. Targeted metabolomics was then used to verify the identified biomarkers. The combination of these methods has allowed for novel insights into pathogenesis of many other diseases, including cardiovascular, neurodegenerative like Alzheimer's, diabetes, chronic disease, endocrine disease, and cancer. These all are chronic diseases, and shaking and shivering the entire mankind from decades together. So what they have mentioned here, recent advance, in order to compensate the pitfall, or in order to mitigate the pitfalls, or in order to overcome the disadvantages, what they have done is, in particular study, hyperuricemic technique, first they have applied untargeted strategy. They have come to conclusion, and then whatever biomarkers they have identified, So they have been verified using targeted, like absolute quantification, targeted strategy. So by combining these things one after other, we can come out with a very good outcome here. So that we can crack, or we can troubleshoot some disease causes like mechanisms, or we can develop some biomarkers, especially in case of Alzheimer's, cancer, diabetes. These are the few diseases, so now we need to pay much attention to. And scientists have used semi-targeted analysis involving hundreds of metabolites, but often without hypothesis. This approach has also provided considerable insights into physiology and disease. For instance, semi-targeted metabolomics played a pivotal role in pinpointing essential metabolites linked to an elevated future risk of pancreatic cancer. This is another case study. Others have integrated metabolomics with genome-wide association studies, GWAS, to establish genetic associations with fluctuating metabolite levels to further understand causal mechanisms underlying physiology and disease. So this is another example for you to generate interest towards this semi-targeted. So there they have combined untargeted, followed targeted. And here in pancreatic disease study, they have gone for semi-targeted. And these are the challenges. I have been telling about these challenges and disadvantages. So big data, how to manage this data. So multi-omics integration, pathway analysis, data mining, database construction, parameter optimization are the few challenges in metabolomic analysis. So here this picture shows the role of each part, like data acquisition, feature extraction, quantification, statistics, annotation, and data sharing and meta-analysis, so and others. So depending upon the nature of the study, sometimes other tools also can be used. So these are the big data challenges.